Hello everyone, welcome to our session on evolution, and today we are going to see evidences of evolution. In the last sessions we discussed about the different theories which is related to the origin of life, and today we are going to discuss about the evidences of evolution. And there are many direct evidences, direct as well as indirect evidences for evolution, and they are as follows. There are the evidences from embryology, evidences from comparative morphology and anatomy, evidences from paleontology, which is from fossils, evidences from comparative physiology and biochemistry, and evidences from taxonomy. So these are considered to be the evidences for evolution to have happened or evolution is happening. So first we will move on to the evidences from embryology. So embryology is nothing but the study of the development of an organism from the egg to the adult. So all organisms, the main evidence for evolution is that every organism they develop from a single cell zygote, which indicate that every organisms are having a single cell ancestors or single cell ancestor. And there is also evidences for similarity in vertebrates or chordates. The similarity in the embryos from fish to man also suggests the evidence for evolution from embryology. And the main evidence is that the ontogeny, that is nothing but the developmental history, that is from the egg to the organism, that is the developmental stages or developmental history from the egg. But after it is getting fertilized to zygote, it is again undergoes cleavage or division, and it slowly develops into a complete organism. That particular stages or that history from an egg to a or that stages from the egg to a organism is called as the ontogeny, and that in every case from fish to man it begins with a fertilized egg, and similar stages are also found in their development. That is, you can see the first cell stage, which is called as a zygote. It divides, it undergoes or it enters a stage which is called as blastula. After that, blastula undergoes further division. There is an increase in the number of cell. Different cells attain different functions, etc. The three germ layers forms, like the ectoderm, which is the outer layer of cells, the middle layer of cells called as the endoderm, and the inner layer of cells called as the mesoderm. So from a single cell, such different patterns or such different structures are being developed, and the same is seen in all these vertebrates from fishes to man, that is taken as an evidence for evolution. Another evidence is the structure which is called as archenteron. Archenteron is nothing but the primitive gut or the primitive alimentary canal, which is starting from the mouth and ending in anus. So the archenteron is the early alimentary canal which is seen in all these groups from fishes to man. So that is also an example, or not example, and evidence for evolution. Next is the fate of the germ layer. So there are three germ layers, mainly the ectoderm, the endoderm, as well as the mesoderm, and all these three germ layers, they're having the similar fate. Similar fate in the sense they give rise to the same type of tissues or the same type of organs. For say the ectoderm, which is the outer layer, which is seen in the embryo, that is the one which is that is the one which give rise to the epidermis, which is the outer layer. Then it give rise to the nervous system, as well as the sense organs. So in every case, in all these different types, you can see that this ectoderm is giving rise to these particular structures. Coming to the endoderm, which is the middlemost layer, which is seen in the gastrula stage, that particular middle layer give rise to the digestive system, which includes the digestive tract, which is the alimentary canal, and the related glands. So that is also the same, or that is seen similar, or the this particular development is seen similar to all these groups. And coming to mesoderm, it give rise to the bones, the kidney, the reproductive system, etc. So all this happens, or all this developmental procedures, or the developmental particular developmental stages or developmental history is same which seen in fishes to mammals, and that is taken as an evidence from embryology. and the presence of the extra embryonic membranes like the the amnion, the chorion, the allantois, etc., which is seen from reptiles to mammals, is also an evidence for a common ancestry for these three groups. That is, the germ layers, the common germ layers are seen from reptiles, birds, as well as mammals. And this germ layer, the presence of the same germ layers in the three groups, suggests they are having a common ancestry. So these are some of the evidences from embryology that suggest, or evidences for evolution, uh, uh, from evidences from embryology. And there are two a principle from von Baer, principles from von Baer, and also the recapitulation theory, which is in support with what is called as our uh, uh, evidence from embryology. So von Baer, he suggested that general characters in an organism appear before special characters appear. So general characters are nothing but, as we said earlier, the formation of the germ layer. So the three germ layers are same in the different groups of animals. That is, the ectoderm, endoderm, mesoderm is the same. The same type of movements are they being exhibited, etc. So that is happening on such general characters are the ones which appear first, and special characters, because every organism does not have the same structures, and such special structures develop later. So that is the principle, that is one of the principle that is laid down by von Baer. Next, more general, that is an explanation of the first point itself. More general characters appear first, after that less general characters appear, and after that finally the specialized character of that particular organism develop. And he suggested that the embryonic stages of an individual will resemble the embryonic stages of its ancestors, in the sense that, the if you take the taxonomy and the embryonic stages of the previous phyla, for say, if you take the embryonic stages of mammal, according to von Baer, he says that the embryonic stages of a mammal will repeat or resemble the embryonic stages of its ancestor, mainly the reptiles, mainly the birds, amphibians, etc., or fishes. So that is what von Baer suggested. But there was a controversy regarding the statement that was put forward by von Baer, and it was later interpreted, or von Baer's principle was later interpreted in such a manner that von Bayer's principle, which is the embryonic stages is resembling the embryonic stages of the ancestor, that was modified into something which is called as a common ancestry. So that is true. That is the von Bayer's principle. Accordingly, if it is interpreted in this manner, it is true. That is, a mammal is having a common ancestry, or these particular organisms. That means the embryonic stages of mammal, a reptile, a bird, a fish, or an amphibian. All these groups are having a common ancestor. That is why their embryonic stages are similar. So that is what is the reinterpreted form of von Bayer's principle, which is widely accepted now. And coming to the second one, which is the recapitulation theory, or the biogenetic law by Ernest Haeckel. So it states that ontogeny recapitulates phylogeny. So ontogeny, as we said earlier, it is the developmental stages from egg to the organism of that is the embryonic development, or the embryonic stages of an organism is called as ontogeny, and phylogeny is the ancestral or the evolutionary history of an organism. So Haeckel postulated that the developmental stages will repeat the ancestral stages of an organism. So the developmental stages, or in this, the higher animals in their developmental stages pass through the adult stages of lower organism, which would be their ancestors. And this is the famous Haeckel's postulation. He says that the developmental stages of the organisms, it is said that they will recapitulate the adult stages of the previous one. That means if you take the developmental stages of any of the group, you can see that if you take the salamander, you can see that it is resembling the embryonic stages is resembling that of an adult fish. If you take the case with chick, you can see that bird, you can see that it is having somewhat the shape of a reptile, and then it forms a bird, etc. So that is what Haeckel suggested, that the embryonic stages of a particular organism will repeat the ancestral stages of that particular organism. Ancestral means the previous phyla, etc. So the Haeckel postulated the recapitulation theory on the basis of these examples. One is tadpole larva, which is of an amphibian. So he says that the tadpole larva, it is being a larval stage, it is repeating the ancestral stages of the or the ancestral stages of this particular amphibian, in the sense that the tadpole larva is having many features that of a fish. That means it is living entirely in water, it has gills, it has fins for swimming, etc., which are the characters of an adult fish. So that is taken as an example, or the tadpole larva, which is a fish-like amphibian larva, is taken as an example for recapitulation theory. Next is the caterpillar larva. So the caterpillar larva, which is of a butterfly, of an arthropod, and it is said that the caterpillar larva, taking its structure, taking its form, etc., it resembles its previous or the phyla, which is the annelidan group. So it has the features of annelida, and that is an example of recapitulation theory. Next is the pharyngeal pouches in all vertebrates. All vertebrates possesses a pharyngeal pouch. Pharyngeal pouch is nothing but the primitive or an embryonic stage of a gill. So that is present in every vertebrates, even man, during the evolutionary or during the embryonic stages, and that in man, because that in mammals, because it does not have any function, it just disappears during the course of embryonic development. So that is these are the three examples that is considered as evidences for evolution, evidences for recapitulation theory, and these two, that is the principle of von Baer and the recapitulation theory, is taken as an examples for evidence for evolution from embryology. And next we are moving on to evidences from comparative morphology and anatomy. Of this, first we will be dealing with homologous organs. So homologous organs are nothing but organism or the organs in seen in different organisms or different group of animals which are having the same structure. Their embryonic structure or the general feature or the general structure basic plan will be the same, but they will be performing different functions. So they will have a structural similarity, but they will be different functionally. And such a type of or such group of organs which are having the same structure but different function is an evidence for something which is called as a common ancestor. They had a common ancestor and they have evolved the particular same structure, but due to their different adaptations or different habitat, it got modified into different forms. Externally they may look similar, but internally they will be the same, or they will have similar structure. And this is said to be due to something which is called as a divergent evolution. That means they had a common ancestor. From that common ancestor, the common ancestor gave rise to the same structures, but due to their function or due to their adaptation to their different habitats, they attained different functions. So examples are shown here. You can see the forelimb of man, the forelimb of a cat, then that of a whale, and that of a bat. So all these are mammals, and you can see that the basic structure of the limb is the same. That means they have put it in the same color. That means similar bones are seen here. That means the bones of the fingers, which are phalanges, which are given in brown. Then that of the humerus or the femur. If it is a hind bone, it is a femur. Then you have the other two bones, the carpels, etc., which is seen shown in yellow, etc. So every basic plan is the same, but you can see that because of the different functions. So the forelimb in man, it is used for grasping, etc. And in this case of cat, it is used for locomotion. In whales, it is used as a flippers, and in bat, it is used for flying. So the functions are different, but you can see that the basic plan is the same. So and the same basic plan suggests that they had a common ancestry, and from that common ancestor they diverged into different structures or different external structures as an adaptation to the new environment, and that is what is called as divergent evolution. And this is what is called as homologous organs. So homologous organs are seen in different organisms or different animals with different functions and same basic plan. And another subdivision of homologous organs is serial homology. So serial homology is nothing but uh, the uh, the same. The definition is the same. That is, they are the organs which are having the same basic plan but seen in the same animal. So that is what is called as serial homology. The example of serial homology or serial homology is homologous organs seen within the same organism when two or more parts of the same organisms have fundamental structural similarity but different function. So the two examples are the appendages of prawn and the hand, as well as the leg bones of the forelimb and hind limb bones of mammals or man. So here you can see that in arthropods, the basic plan of the appendage is the same. That is, they are having a biramous plan of appendages with the two branches, the exopodite, endopodite, and a protopodite with coxa, basis, etc. But you can see that as their function changes. For example, take the case of the antenna or the antennule. They are having the same biramous pattern of structure, but because of their difference in their function, they are having different structures. For example, the mandibles, which are meant for chewing, they have the same basic structure, but they are modified only for chewing. The structure, the shape, etc. changes. The same is the case with the walking legs as well as the swimming legs. You can see they also are having the same biramous pattern. They have the exopodite, endopodite, but the size, the shape of the exopodite, the size of the exopodite, the number of segments, etc., is different due to their different function. Here they are used for walking, and these are used for swimming, etc. So serial homology is nothing but the homologous organ seen in the same organism. Here the same organism is having the same or the same structural features, but their function is different. The same is the case with the forelimb and hind limb. You can see that a similar type of bones are seen in the forelimb as well as the hind limb. In forelimb, you can see the humerus. In the hind limb, you can see the femur. You have the radioulna. Here it is the tibia fibulae. You have the carpals. Here the tarsals. Then here the phalanges. Here also you have the phalanges or the finger. So the basic plan is the same, but the function is different. This is used for locomotion, and this is used for other purposes. So this the particular type of homology which is seen within the same organism, that is called as serial homology. Coming to the next type of uh, uh, organs which is seen, which is called as, which is also an evidence for evolution, which is analogous organs or analogy. So here the organs differ in the embryonic origin. They do not have the same pattern or same structure, but they have the same function, and this is because of the adaptation to the similar mode of life or similar habitat. So analogy is similarity due to function, and even though they differ anatomically, and this is a resultant of something which is called as the opposite of homology. Homology was due to divergent evolution, and this is due to convergent evolution, convergent to a basic function. So analogy is not due to a common ancestry, but adaptation to similar habitat. So the examples are shown here. It is the wings of insects, the wings of bird you can see, and the wings of bat is also given. So in every case, you can see that this everything is used for the same function. It is for flying, but the basic structure is different. This is made up just of chitin, which is arthropodin. Here in the case of bird, you can see that this is made up of bones, then feathers which are covering it, etc. So here in the case of mammal, which is bat, you can see the same pentadactyl plan. Here it is the flying reptile. Here also you can see that it is made up of bones, and the skin is being spread to assist the function of flight. So all these, they differ structurally, but they have the same, they do the same function, and that is why they are called as analogous organs, and this particular phenomenon is called as analogy. Next is vestigial organs. They are remnant organs which are found, which were functional in the ancestors, but that is present in the organism that you see now, but they do not have any function now. Such organs are referred to as vestigial organs or vestiges. So this is because of the changed environment. In the changed environment, in their changed habit, their changed food habits, etc., they become non-functional. Such structures which are even present now without any function are called as vestigial organs. The examples are the vermiform appendix in man. So vermiform appendix is a particular structure, a particular tubular structure that is seen at the junction of the large intestine and small intestine, and this is considered to be a vestigial organ. So this particular structure, it is now a reduced structure, but it was a functional structure when the ancestors were completely herbivore. So in herbivores, you know that the intestine is very lengthened because of their particular type of habit and the particular type of digestion they are having. So when evolution happened, or when humans evolved, what happened is when their habitat or habit changed, what happened is this didn't have any function. The vermiform appendix, the portion didn't have any function. As a result, it became functionless, but its remnants are seen. Only the remnants will be seen, not the entire structure. The remainings, or a piece of such structures, will be seen, and that is what is called as the vestigial organs. The same as with the case with coccyx, that is a tailbone which is present in humans. This tailbone, it is seen at the terminal region of the vertebral column. So here also you can see that the coccyx bone, it doesn't have any function because we do not have a tail. As a result, this particular structure doesn't have any function, but they are present in man because the extensions of the tailbone is what is called as a tail, but as tail is absent, a small piece of the coccyx bone is present, and that is that is considered to be vestigial. And there are many vestigial organs in man, like the pointed canines, then the wisdom tooth, which is a third molar, vermiform appendix. Then you have the caudal vertebra, said body, the hairs on the body. Then mammae on mammals, it does not have any function in males. Then you have the muscles of to move the ears. We have the muscles, but we can't move them because they are vestigial. Next is the nicticating membrane, that is also the vestigial organ, that is vestigial structures. So these are some of the examples of the vestigial structures that is seen in man. Next is atavism or reversal, that is the reappearance of the ancestral characters after interval of many generation. Example is the cervical fistula and profuse hair growth. So this is a profuse hair growth, and this is also an example, that is the tail. The extension of the coccyx bone to form a tail is considered to be atavism. Cervical fistula is nothing but the presence of the pharyngeal gill slits. So as I said earlier, pharyngeal gill slits are seen or also seen in human embryos during their early embryonic stages. In this, they are paired, and this seen in the throat region. So what happens is when our lungs are formed and it starts, or when in the stages of development, what happens is when a functional lungs etc. is developed, this the particular structures which were like holes which were seen in both the sides in paired forms, it just closes off. They will not be seen in the adult stages. But when you come to atavism, what happens is when the particular baby is born, it will have holes in this particular sites, and that is what is called as cervical fistula, which is not closed in a normal procedure, and that is a reversal of what is called as the ancestral structures or ancestral characters, and that is what is called as atavism. So next is the last one in this particular group, which is the connecting link. So the animals exhibiting the characters of two adjacent phyla are called as connecting link. They represents the transitional stage of two groups. You might have studied the examples earlier, like peripatus, which is a connecting link between Annelida and Arthropoda, and Protothera, which is a connecting link between the egg-laying forms as well as the mammals. They are the egg-laying mammals, so they are the connecting link between the oviparous as well as the viviparous organisms. So that's all about the two evidences for evolution. So today in evidences of evolution, we have come across evidences from embryology, where we saw von Bayer's principles and the biogenetic law, or Haeckel's recapitulation theory, and we also saw the evidences from comparative morphology and anatomy, that is where we saw homology, analogy, then vestigial organs, connecting links, as well as atavism. And the rest, paleontology, comparative physiology, we'll deal with this in the next session. So thanks for hearing.